Okay, good afternoon, everyone. The NSRI lecture series will begin in 5 minutes. Please be informed that this webinar is being recorded and live streamed in YouTube. All participants are reminded to keep their microphones and video cameras off during the program, and please use the chat box for questions and clarifications only. Now, those watching via YouTube may submit their questions through the Google form indicated in the YouTube page, and those who would like to receive a certificate of attendance are required to accomplish an evaluation form through a link that will be posted in the chat box later in the program. Thank you. Okay, so good morning once again. This is the NSRI lecture series for the Science, Technology, and Society Month for 2020. Thank you for joining us. The theme for today's event is Genomic and Molecular Tools for Biosurveillance of Bacteria and Parasites. My name is Hilpin Liar. I'm from the Institute of Biology, and I will be your host and moderator. Now, please be informed that this webinar is being recorded and streamed live in YouTube. All participants are reminded to keep their microphones and video cameras off during the program, and please use the chat box for questions and clarifications only. Now, those watching via YouTube may submit their questions through the Google form indicated in the YouTube page, and those who would like to receive a certificate of attendance are required to accomplish an evaluation form through a link that will be posted in the chat box later in the program. Now, this afternoon, we are very fortunate to have three excellent researchers from the Natural Sciences Research Institute who are also faculty members of the Institute of Biology. Now, before we continue, I would like to acknowledge our registered participants from the academe, research, medical, industry, and other sectors. Now, thank you very much for joining us today. Now, to formally open today's program, I would like to give the screen to the Acting Director of NSRI, Dr. Jonas Kilang, for the welcome remarks.
Hello, everyone. Good afternoon. On behalf of the Executive Council, scientists, researchers, and staff of the Natural Sciences Research Institute, I'd like to welcome you all to the first installment of the NSRI webinar series. The theme for this afternoon's webinar is Genomic and Molecular Tools for Biosurveillance of Bacteria and Parasites. Although we released the announcement poster for this webinar barely 10 days ago, we still got a huge number of participants, over 600, who signified their interest to attend this webinar. We have participants from all over the country. We are very grateful and we are honored to have with us today two former directors of the NSRI and the current director of the Institute of Biology, UP Diliman, as our speakers. They will be presenting the output of their research that was funded through a grant obtained by the NSRI from the Department of Science and Technology, Philippine Council for Industry, Energy, and Emerging Technology, Research and Development, or PCERD. As the National Center of Excellence in the Natural Sciences, the NSRI is mandated not only to undertake research in biology, chemistry, mathematics, material science, environmental science, and meteorology, but also to disseminate research findings through scientific publications, workshops, conferences, seminars, and now webinars in this time of pandemic. Now, the NSRI is also mandated to provide extension and other services and analytical support to students, researchers, other units of the university, and other institutions and agencies, both government and private. We have four in-house laboratories at NSRI: the Biological Research and Services Laboratory, the DNA Analysis Laboratory, the Microbiological Research and Services Laboratory, and the Research and Analytical Services Laboratory. You may refer to our website or Facebook page for more details on the research activities and extension services of our four in-house laboratories. Now, before I end my opening remarks, I'd like to thank the speakers, the moderator, the NSRI Seminar Committee, and to all of our attendees for being here with us today. To all the attendees, I hope that you will be able to pick up new knowledge from the three lectures. For the biologists, microbiologists, parasitologists, and geneticists among the audience, I hope that you will be able to get ideas on how you can apply genomic and molecular tools to your own research. To the senior high school and college students and teachers, and to the general public, I hope that you will be able to gain a better understanding and appreciation of how bacteria and parasites can be detected and monitored using molecular tools. So again, from all of us at NSRI, welcome to our first webinar and thank you.
Thank you very much, Dr. Kilang. We will now proceed with our webinar. Now, our first speaker is Dr. Wendell Rivera. He obtained his PhD in medical science from Nagasaki University. He is a Professor 12 and a UP Scientist 3. He's an academician of the National Academy of Science and Technology and the current chair of the Philippine Academy of Microbiology Council of Regents. He's a former director of NSRI and the current principal investigator of the Pathogen Host Environment Interactions Research Laboratory to talk about Aptamer-based assay for the detection of Trichomonas vaginalis. Let us all welcome Dr. Wendell Rivera.
Thank you very much, Dr. Pinulyar, for the introduction. And of course, thank you very much, Dr. Kilang, for inviting me to give this lecture this afternoon. Okay, so let me just share my screen. Okay, can you now see my slides?
Yes, sir.
Okay. So this afternoon, I'll be discussing about aptamer-based assay for the detection of Trichomonas vaginalis. So just to give you an outline of my presentation, I'll be starting with the discussion of the organism of interest in this lecture, that is Trichomonas vaginalis. Then we'll go on discussing what aptamers are and the aptamer that we have developed for this particular study. We called it AP65 aptamer. So for those of you who have a background in parasitology, I am sure that you well know what Trichomonas vaginalis is and its biology. So from this structure, clearly we can see here that the organism is of the shape, almost ovoid. It has 4 flagella protruding from the anterior part of the cell. And there's also this unique flagellum, and we call it undulating membrane, also coming from the anterior part of the cell. Of course, it has its nucleus, and another interesting structure in Trichomonas vaginalis is the presence of axostyle, which is basically a rigid structure that protrudes from the posterior end of the parasite. Now, this is an interesting picture of how Trichomonas vaginalis adhere to epithelial cells, but in particular, these are known human vaginal epithelial cells. So primarily, we can see here tropism, that the organism basically adhere to human vaginal epithelial cells, causing trichomoniasis. So as to motility, this is a typical representation of the jerky movement of the cell, which is typical of any flagellated organism. It is quite stable in room temperature, and if you want to put it in a solution, say for example phosphate buffered saline, it is quite stable in PBS for about 6 hours, but eventually it attenuates, okay, and it stops, of course, and it may die. Okay, so basically this is a photomicrograph of a wet mount. So if you are into diagnosis of this parasite, you can clearly use this as a diagnostic procedure. Well, the disease that is caused by the organism is known as trichomoniasis. And as you can see here, it basically infects both the urogenital tract of men and women. At times, it may cause respiratory tract infection, which is basically due to some sexual practices of individuals, like orogenital sexual practice. But there are also cases of trichomoniasis in neonates, and due to the passage of the organism through the birth canal, the neonate is able to get respiratory tract infection as well. Now, as to the pathogenic mechanism of the organism, say, for example, this is the cell itself that invades the host cells. Okay, it may incite inflammation, which eventually may subvert the host immunity. At the same time, it may cause damage to host epithelia, just like the slide that you have seen earlier of the trichomonas adhering to the host epithelial vaginal cells. And it may also cause disruption in vaginal microbiota. And this is known to be dysbiosis, which is very typical to many pathogenic protozoan parasites. Now let's move on to symptoms and complications of the disease. In men, the most typical symptoms would be urethritis, epididymitis, prostatitis, and also infertility. In women, it may cause frothy, malodorous discharge, abdominal pain, vaginitis, and signs of strawberry cervix. Now, in neonates, okay, so we have seen earlier, it may also cause low birth weight for the infants, preterm birth, and as what we have discussed earlier, respiratory failure also. Now, interestingly, this parasite, majority of the infection is asymptomatic. About 70 to 85% of infected individuals have minimal or no symptoms at all. But then again, studies have shown that this infection may cause 50% increase in the risk of HIV-1 acquisition. Well, there are many factors why we can say this, one of which is the inflammation that I have described earlier in the human epithelial cells, vaginal epithelial cells. There is basically a recruitment of CD4 plus on that area. And as we know, HIV-1 targets the CD4 plus T cells. So because of that, there's an increase in the risk of HIV-1 infection. Of course, there are also studies showing that the parasite is able to resist drugs like metronidazole and also tinidazole. Now, WHO gave us an estimate, and this is quite old, in 2008. We don't have a more recent data yet. But as you can see from the pie graph, majority of the infection is due to the Trichomonas vaginalis for sexually transmitted infections. So it's the most prevalent non-viral curable sexually transmitted infection. It is considered as a non-reportable disease. And one of the CDC's 5 neglected parasitic infections, along with Chagas disease, cysticercosis, toxopariasis, and toxoplasmosis. Now let's move on to the current detection assays being used for the detection of the parasite. Well, there are basically 3 methods, one of which is wet mount or culture. This is the most traditional, and yet this is still used as the gold standard for detection of the parasite. Okay, another one is nucleic acid-based test. Of course, in the advent of molecular biology techniques, we now have a lot of probes that are used for detection of the parasite. And of course, antibody-based assay. Now, let us try to compare and contrast these 3 methods, okay, as to sensitivity, specificity, turnaround time, and also some issues. Okay, clearly we can see in the wet mount that it has low sensitivity but higher specificity. But that is basically due to this factor, which is technician dependent. So it basically is dependent on the skill of the microscopist. Now, nucleic acid-based test, on the other hand, has higher sensitivity compared to wet mount, almost similar specificity, but a shorter period of time for the detection process. But normally, this is expensive due to the equipment that are being used and, of course, the reagents. Now, the third one, antibody-based assay, is also comparable to the nucleic acid-based test. In fact, but in the sense that it has almost the same sensitivity and also specificity, a slower, faster turnaround time. But this is the issue or concern in this particular method. It is expensive. And of course, you know that it's really difficult to produce antibodies. Now, this is a kit that is produced by Sekosei Diagnostics of Japan. And this is basically the sole true point of care assay available in the market today. It's an immunochromatographic detection assay. It is quite expensive in the sense that one kit can actually test around 25 individuals, and a kit would cost around 434 US dollars. And so if we compute, one test would cost 17.36 US dollars. That is nearly $20. So it's about 1,000 pesos per test. But of course, we know that according to WHO, when we talk about point of care criteria, we actually mean these characteristics. It should be affordable, sensitive, specific, user-friendly, rapid and robust, equipment-free, delivered to those who need it. So that is the abbreviation of uh, assured. Okay. Now, is it really affordable? Well, from the price that I have mentioned earlier, it seems that it's not. And is it delivered to those who need it? Well, we can say no as well, because we have many cases of trichomoniasis in the country, but unfortunately, they're not able to access this POC detection kit. Okay, now let's move on to our discussion of the aptamer. What are aptamers? Okay, probably you've heard this. This was first developed in the early 1990s. Okay, and it's a synthetic. It's known to be a synthetic antibody, meaning that it can be an alternative biorecognition molecule. So it can detect small molecules. It can detect your proteins. Okay, of course, it can also detect cells. So just like your antibodies. Okay, it came from these two words, one from Latin expression aptus, which means to fit, and Greek word meros, which means part. Okay, so aptamers are basically artificial nucleic acids, whether they are RNA or DNA. Mostly they are single-stranded, but with specific three-dimensional shape. Now that specific three-dimensional shape is quite important in the binding. Okay. To the target molecule. Okay, so you have a folding, of course, that would happen from your RNA or single-stranded DNA. Okay, and binding is due to this conformation, the three-dimensional structure of the nucleic acid. Now, it binds to the target complex, and they are described to be induced fit. It has structural compatibility. Electrostatic interactions are involved, and as well as hydrogen bridges. Now, what are the advantages of using DNA oligos over antibodies? Okay, first and foremost, DNA oligos are chemically synthesized. There's no batch-to-batch variation, okay, which is quite different if you are producing antibodies. It is advantageous over all kinds of molecules, as I said earlier. The small molecules, your proteins and cells can be targeted by aptamers. Okay, there's an ease of modification and high flexibility, and this is quite difficult in antibodies, because if you try to modify, the function may also be modified as well. And of course, being DNA oligos, it's highly stable at room temperature compared to this protein molecule antibody. And there's also reversible denaturation. Now, to actually produce or select aptamers, as you can see in this slide, it seems that it's quite complex method, but it's actually not. All you need to do is to begin with a DNA library. So you would have different sequences of DNA here. It's a pool of library. It's random, okay? And you basically incubate it with your target molecule, okay? So once you incubate it, of course, you expect that those with specificity to the target would bind. Okay, so this is the first step in the selection process. And by the way, the selection process is known as SELEX, short for selective evolution of ligands by exponential enrichment. Okay, once the aptamer is bound to the target, clearly you need to, of course, separate those that did not bind to the target molecule and you have to separate them from those who from those which bound actually bound to the target molecule. Okay, so you call this a negative process because you're trying to remove those that did not bind to the target molecule. But of course, you need to elute those that are bound to the target molecule. So to elute that, you have to specifically target that those DNA molecules or aptamers that bound to the target molecule. Now, this is a continuous process, as you can see here. It's a cycle. So this is one round of select procedure, and you continually do this, okay, for you to be able to enrich the library. Now, in the selection process, as I mentioned, there are three types basically. One is negative selection. I mentioned this is basically the part of that negative selection. You are trying to remove those that did not bind to the target molecule. And most likely, these are the ones that bound to the matrix of your platform. Now, the second selection is counter selection. Basically, this is a removal of molecules that are target related. So they are similar in structure, probably. They bound to that specific molecule, but that is basically not the target. So you have to remove them from the process. Now, the last one is positive selection. Basically, this is the last part of that. You are able to elute those aptamers that are very specific to the target, okay, and you are able to amplify it and characterize it as well. So the goal of this research was to explore the potential of aptamers as the molecular recognition element in the development of diagnostic tools for T. vaginalis detection. So the procedure is basically starts with aptamer selection. Okay, we actually use a microtiter plate in this process. Okay, followed by aptamer binding affinity studies. Okay, using these methods, which I will discuss later on. Specificity studies, of course, this is quite important for us to characterize if the aptamers that we have developed are really specific to the target molecule. And in this particular study, we also performed dual aptamer enzyme-linked aptamer assay using recombinant AP65 protein, which is basically the target molecule for this study, and using T. vaginalis cells. And this is our hope to be able to produce an aptamer-based lateral flow assay for Trichomonas vaginalis infections. Now, interestingly, the first three parts of this project is already published in ACS Infectious Diseases. So we are currently working on these last two aspects of the project. Okay. Now, the target molecule that I have been mentioning is AP65. It's basically an adhesion protein. So if you remember that slide earlier showing us the adhesion of the parasite to the host vaginal epithelial cells, this is basically the molecule that is responsible to that adhesion. So it's a dual dimer, okay, and it's known to be a hydrogenosomal malic enzyme regulated by iron. It's a prominent adhesion associated on the parasite cell surface and is secreted to the extracellular environment. So please take note of this characteristic as we will try to discuss this again later on. So it's an adhesion that is found on the parasite cell surface, but they can be secreted extracellularly. Okay, and lastly, they play a role also in the adhesion of the parasite to host cell and to iron-rich heme and hemoglobin. Now, I have discussed this earlier, and this is basically the enrichment part. Okay, we perform this basic procedure several times, a number of rounds. In this particular study, we've used around 8 rounds. And then after we have enriched the oligos, we subjected them to next generation sequencing for us to be able to characterize the aptamers that we have developed. We used microplate-based SELEX assay in this particular study, but traditionally, magnetic beads are actually used. Okay, now why did we use microplate-based SELEX? Well, primarily because of these 4 advantages. One, because of its small-scale preparation of freshly thawed target for immobilization, easy confirmation of target immobilization through ELISA, easier removal and washing of the unbound oligonucleotides. For you who have actually done ELISA, you can clearly relate to this advantage. of how to remove and wash unbound molecules. And last advantage is its less matrix interaction. Magnetic beads contain more matrix components, which can actually lead to a less specific aptamer. Okay, now this graph actually show us two types of bars. One with a faded gray color bar, and you have these dark colored bars. Okay, so I mentioned about SELEX that refers to a selection process, and it has to be done in different rounds, in a number of rounds. So the selection rounds here shows us around 1 to 8 with positive and negative selection. Okay, so in the first part, we call it pre-round SELEX. Okay, if you are to run a gel, you can clearly see that you have many of the unbound molecules here. Okay, or the target, sorry, the unbound aptamers, okay, on the wells. So they are actually bound to the matrix, but they did not bind to the target molecule. So you have less of the positive wells in this graph, as you can see. But after round 4, you have more now, you have more concentration of those that bind to the target cells and less of those that bind to the matrix. So clearly we can see here the evolution of the aptamers. Okay, after round 6, it became higher, but of course, due to some reasons, of course, we may have a lower concentration. But this is a good indication that we have actually amplified and enriched the oligos that we need for aptamer selection. Now, I mentioned that after we have enriched the oligos, we need to subject them to next generation sequencing for us to be able to characterize the aptamers that are produced. So in the NGS, we are able to predict the structure using the MFold software. And we were able to identify 6 full-length AP65 aptamers. We call them A1, A2, A3, A4, A5, and A6. These are basically 695 more unique and most abundant DNA sequences that bind to the target molecule AP65. Now, after determining those structures, it's also important for us to get these two important approaches. One is for us to get the G-score and also the truncation process. Okay, G-score is basically a statistical parameter for predicting the probability of finding the G quadruplex motif. Now, this G quadruplex motif is very important because it would show higher stability and resistance over unstructured sequences. So if you have more of this G quadruplex motif in your aptamer, selected aptamer, the more that it will be more stable and it will bind specifically to the target molecule. Now, another factor, this is an example of an aptamer, but this one is directed to thrombin. Okay, clearly we can see loops, at times which we see hairpin structures. Now, the truncation process is also important in aptamer production because when we say we truncate, okay, we actually delete non-essential nucleotides. Now, where do we find those non-essential nucleotides? This can be part of the constant region of the 3' or the 5' end of the aptamer. So you don't need them much, but you should concentrate on the G quadruplex motif. So you have to basically get those with conserved G quadruplex motif. Now, the truncated aptamers now will have higher affinity and specificity for its target. So I mentioned earlier that we were able to produce 6 full-length aptamers, but we, aside from those 6 full-length aptamers, we were able to add truncated aptamers as well. So this one is a truncated aptamer of A1 with a G-score of 21 and has a 14 nucleotide derivative. This is A2 with a G-score of 20 with a 26 nucleotide derivative. A3 with a G-score of 21 and a 20-mer derivative. Interestingly, we did not get a good score, G-score for A4 as well as A5, but we got a good result for A6, which has a 24-mer nucleotide derivative. So clearly from this slide, we have additional 4 truncated aptamers. Uh, if we add the 6 full length, we now have 10 candidate aptamers. So the next procedure is to perform these aptamer binding affinity studies, and there are a number of methods by which we can do this. And this one was not done in NSRI, but this was done in our collaborative partner in Spain. And the method is known as surface plasmon resonance. This is a good tool for characterization of biomolecular interactions. So basically, this is optical based, and you need to have a gold sheet, okay, to immobilize the target molecule, and you run through the aptamer here, and eventually it will give you data on the binding ability of the aptamer. Another method is biolayer interferometry, which is basically similar to SPR, but this one uses biotinylated oligonucleotide, which is of course detected eventually by an enzyme and a substrate. Okay, but probably you're more familiar with ELISA. So ELISA is basically similar to ELISA. ELISA is enzyme-linked aptamer assay. So you basically bind the target molecule onto a matrix, recognize it using an aptamer biotin and a signaling molecule, streptavidin peroxidase, eventually read by a substrate. Okay, so to characterize the binding ability of the aptamer to the target molecule, it's important that we understand the dissociation constant. Okay, so from your chemistry, this is quite important if you are characterizing affinity of biomolecular interactions. So we did 2 platforms. One is that the 8065 target molecule was the one that is immobilized on the surface matrix. The other platform was the biotinylated aptamer was the one that was immobilized on the surface or on the matrix. And we characterized them further using the methods that I described earlier. So what we got was this KD or dissociation constant of 0.965 using ELAA, and there's also 3.21 using SPR. There are differences, of course, in the values, primarily because of the immobilization strategies that we used. Absorption is involved in ELAA, while cross-linking is actually involved in SPR. Further, when we characterize this target molecule as well as the bound aptamer, we were able to get this KD of 209 nanomolars and SPR of 0.577. Now, clearly you can see that in this, there are 2 platforms used, methods, okay, but we use the same method for checking binding ability. But interestingly, we found differing values for KD, like 3.21 here in this case, and 0.577 in the case of the aptamer being bound to the surface. So clearly, aptamers may have different binding affinities depending on its conformation, immobilized or free in solution. Well, the 3D conformation that I mentioned at the outset, it clearly shows that, of course, this one is quite free in solution. It's able to fold in its 3D structure, but this is more difficult for the aptamer, which is immobilized onto the matrix. So that basically explains the difference of these values. Okay. And when we further characterize all the full-length aptamers, we were able to see that the A1, which has the lowest KD, is actually the one that binds well with the target molecule. So take note that the lower the KD, the higher is the binding ability of this molecule to the target molecule. Okay, so when we try to compare the aptamer that we have produced over activity of the polyclonal antibody to the same molecule, AP65, we have seen that there's higher affinity, okay, for the aptamer over the polyclonal antibody, and the same is true with this graph. Okay, so now let's move to the third one, which is specificity studies, and we basically use SPR and ELAA for this assay. Now, clearly from this, we have seen that the aptamers we produced did not get a good signal when we use control proteins like bovine serum albumin or streptavidin. So basically, it's very specific to the AP65 molecule target. So the same is true for this graph when we used ELAA. Okay, so using indirect ELAA and the aptamer that we have produced, clearly we have seen here that the absorbance is high, meaning to say that the activity is quite good, okay, with regard to targeting the molecule. But we have seen minimal cross-reactivity to a Neisseria gonorrhea, the gonorrhea-causing organism. But the possible competition here is maybe due to the biotinylated aptamer that we used for indirect ELAA, and they were able to detect the cell's endogenous biotin of Neisseria gonorrhea. But of course, we need to solve that problem. And to solve that, let's move on to the fourth step, that is dual aptamer ELAA. When we say dual aptamer, we may basically detect the recombinant target, the recombinant AP65 of the T. vaginalis cells or the T. vaginalis cells themselves. Okay, so this is the platform that we used for dual ELAA. Okay, we basically use a capture probe, which is a thiolated aptamer. This is the target molecule AP65, and a reporter probe, which is basically a biotinylated aptamer. It was recognized by the signaling molecules streptavidin with HRP, and the substrate is TMB. Now, the target used in this particular assay were basically the recombinant AP65, as I said, or the cells, Trichomonas vaginalis cells. So remember I mentioned earlier that we were able to produce 10 aptamers. There are 6 full-length aptamers and 4 truncated aptamers. So if we make combinations of all of these 10s, we have 100 combinations, and we tested all of these 100 combinations in our assays. And interestingly, we found this data. This LOD is limit of detection. Of course, the smaller LOD, it means that you have a better assay. So we used A6 here as a capture reporter using the truncated aptamers. And we found that these are the top 3 candidate aptamers for the dual assay. So this one uses the AP65 molecule. Now, interestingly, we did not identify these same aptamers, dual aptamers, for when we tried to use Trichomonas cells. So we have different top 3s. Now, why the difference? Well, primarily because of the target molecules that we have used. Remember, I mentioned earlier that AP65 is found on the surface of the cell. They can cause adhesion. But of course, AP65, in our recombinant AP65 molecule, is highly available to the target, to the capture and reporter aptamers. In contrast, these are live cells. These are cells that we have actually used to associate them with aptamers. And aptamers can be secreted out of the cell, and they are also membrane bound. So it's not that easy for the aptamer to identify them when you use Trichomonas cells. So that basically explains this table. Okay. Interestingly, we also use this assay, aptamer antibody assay. So there's an aptamer as a capture. Rio reporter is basically an antibody molecule, or the other way around. You have an antibody capture and reporter aptamer. So you can actually mix several combinations in your sandwich ELAA. And we were able to get this data for polyclonal antibody that were used as a reporter and a polyclonal antibody that was used as a capture molecule. Okay, so there are also differences, but we targeted this one because it ranked 1 here and it ranked 2 on this LOD concentration. So A6 is a good candidate for targeting AP65. Again, just to go back, we have clearly identified as well these molecules that identified around, this one is the lowest, 414 cells. The second one is 472 cells, and the third one, 489 cells. So again, because of their lower limit of detection, we can say that they are good candidates for aptamer that are specific to the Trichomonas vaginalis cells. Okay. Of course, normally you find the parasite in different biological fluids. So we try to check. These are simulated biological fluids like urine, semen, vaginal fluid, and as a control, we use PBS. But from these two graphs, using the full-length aptamer and the truncated aptamer, clearly we can see that when we use PBS as a binding and a sample buffer, it gave us a good binding ability, okay, compared to the others. No? But still, we can say that we are able to detect the parasite coming from these biological fluids. Okay, so let's now move to the last one, the last part of the study, that is aptamer-based lateral flow assay. Okay, this is the platform that we are thinking to actually work on and basically produce later on as a product of the development of an aptamer that is specific to Trichomonas vaginalis. It's a lateral flow assay, and we would like it to be paper-based. And for Reporter Probe, we are interested in producing aptamer functionalized gold nanoparticles. And we were able to successfully conjugate them to the aptamers that we produce. So this is the gold nanoparticle conjugated to the full-length aptamer as a reporter probe. The other one is a gold nanoparticle conjugated to the 24-mer. This is a truncated aptamer, and they also gave good signals. So hopefully, eventually, we'll be able to mainly produce these platforms for a good point of care assay for Trichomonas vaginalis infections. So let us summarize the data that we have generated from this study. We have seen that 10 aptamers are quite successful in getting a good binding to the target AP65. It binds to AP65 in solution, that is the recombinant protein, and also on cells when we tried them on Trichomonas vaginalis cells. We also found that it can bind simultaneously to the target. It can be paired with antibody, and it can be conjugated to gold nanoparticles for our DREAM aptamer-based lateral flow assay. Thank you very much, everyone, for listening. I would like to thank, of course, our collaborator from Tarragona, Spain, Universidad Rovira Virgili. The group is known as Nanobiotechnology and Bioanalysis Group. And, of course, PShared NSRI for partly funding this project. I would like to specifically mention the names of the whole team. Dr. Christian Adam Espiritu, who got his PhD in chemical engineering based on this study. Christine Aubrey Justo, who got her MS Microbiology also from this project. Dr. Annalisa Puyol-Leon of the College of Engineering. And our collaborators from URV, Drs. Rubio, Isbovot-Dova, and Kiras O'Sullivan. Thank you very much once again, everyone.
Thank you very much, Dr. Rivera. May I there. So I'll just share the rest of the screen. Okay. So thank you very much, Dr. Rivera. So as mentioned during the talk, Trichomonas is a significant STI in the world. So there's actually a need for the rapid detection of the parasite, so that the infection it causes can be managed later on, and the identification of aptamers which can bind to specific targets in T. vaginalis is a great step towards developing kits for their rapid detection. Now, we will have the open forum for this talk after our third speaker. But if you have questions or clarifications for Dr. Rivera, you may write them in the chat box or Google form for those watching via YouTube. Now, for data privacy, all questions submitted for the open forum will be considered anonymous unless you type your name before the question. Now, to continue, our second speaker is Dr. Ernilea Cao. She obtained her PhD in biology in UP Diliman. She is a Professor 12 and a UP Scientist 1. She was a former director of the Institute of Biology, a former Deputy Executive Director of the Philippine Genome Center, and a former Director of NSRI as well. Currently, she is the Principal Investigator of the Plant Genetics and Cyanobacterial Biotechnology Laboratory, and to talk about the metagenomic sequencing of cyanobacteria from some mining sites in Benguet Province for genome mining of stress tolerance genes. Let us all welcome Dr. Ernelea Cao.
Hello, good afternoon, everyone. First, I'd like to thank the NSRI for inviting me to present this talk, which is about our work that is part of the research program funded by UP-NSRI and DOSTP Shared. Let me share my screen first. Okay. Can you see my slides?
Yes, ma'am. Thank you.
All right. Thank you. So as mentioned by our moderator, thank you for introducing me, Dr. Gil Penolyar. The title of my talk is Metagenomic Sequencing of Cyanobacteria from some mining sites in Benguet Province for genome mining of stress tolerance genes. So my organism of interest would be the cyanobacteria. And for those who are not familiar with this group of organisms, they are also referred to as the blue-green algae. And they are made up of certain pigments, particularly phycocyanin, which gives the organisms their blue-green color, as well as chlorophylls that allow them to become photosynthetic prokaryotes. They are ubiquitous in nature, found in a variety of environments from marine, freshwater, and terrestrial habitats, and they occur in different forms. They occur singly or in colonies or in filaments, and they are said to reproduce asexually by fragmentation or by fission. And their locomotion is through the gliding movement due to the slime secretion along with contractile waves on the cells. Now, why did we conduct this kind of project? Well, cyanobacteria or the blue-green algae have been reported to survive in a variety of environmental conditions, even extreme environments. And characterization of genes involved in tolerance to environmental stresses, particularly metal stresses, has not yet been done, especially for local isolates. And genome mining offers a rapid approach to the identification and elucidation of genes and gene clusters associated with traits of interest. So for this particular study, my trait of interest would be metal tolerance or metal resistance. And this time I did a different approach. So the usual approach that we would do would be to collect water samples and then try to isolate and purify different strains of cyanobacteria and characterize them in the laboratory, subject them, let's say, to different metal stresses, different kinds of metals, different concentrations, and determine which isolate will be able to survive, and therefore see their potential for bioremediation. At the same time, then we can explore the genetic basis of such metal tolerance. But this time, so what I did is what I call a top-down approach. So I wanted to scan the metal contaminated environments and determine the kinds of cyanobacteria that can survive and determine the genetic basis for such ability. This is where the genome mining came in. So the objectives of the study were the following: to generate sequences of cyanobacteria from some mining sites in Benguet Province using metagenomic sequencing, to determine the diversity of cyanobacteria present in the said environment, and to mine putative gene clusters that may be responsible for some stress adaptations like metal tolerance. So for those who are not familiar with this technique, so metagenomic sequencing refers to the sequencing of the entire genomes of all microbes present in a sample to explore their taxonomic, functional, and evolutionary aspects. This is also referred to as microbial community genomics. And as you probably have noted, then this will be culture independent. So let's say in a particular environment, as shown here in this picture from one of our sampling sites, so we can sequence all the genomes of the microbes that would be present in the water that would be found in this mining site. So for the particular materials and methods that we followed, these are the following. So we collected for water samples, particularly surface water, because we were interested in cyanobacteria, and they would predominate on the surface because they are photosynthetic. They will seek the light. And we went to Benguet for our sampling because this will be where a number of mining sites are located. So in the town of Itogon, we actually collected from the Balatok mines. This is actually a big mining site, the first actually and the oldest mining site in the area. And however, this was already, or this is already abandoned since 1993. So I wanted to find out if there are still cyanobacteria that we can collect from this sample. And then we also collected from a smaller mining site in Akupan. And we went further up in Tuba Benguet and also made collections in a smaller mining site called the Ampukao mines, and then went to a very big mining site owned by a large mining corporation, the Philex Mining Corporation. And in there, they had two sites, the inactive site, which had already been revegetated, and an active site. So we collected in both and actually conducted sampling in each site at 3 sampling points, and brought this to the laboratory for DNA extraction. However, due to limitations in budget, because high throughput metagenomic sequencing is still quite expensive, we were only able to allot budget for the sequencing of 9 samples. So what I did was to choose a representative from the town of Itogon. So I chose the Balatok sample representing the abandoned mining area. And then the one in Tuba Benguet, the Felix mining sites, the inactive and the active mining areas. Note that we collected from the tailing ponds because this would be where the extra metals will be discharged from the main mining facility. And eventually, the promising cyanobacteria were cultured and maintained in the laboratory. So some details on the sampling that we conducted. So we also did some on-site characterization of the areas where we sampled. So we got the GPS coordinates. We brought a portable multi-parameter probe that measured the temperature, the total dissolved solids, as well as salinity and conductivity of the water. And using a portable pH meter, we also got the pH of the water. And so as I was mentioning, we randomly sampled from 3 sampling points. The revegetated inactive site is a relatively smaller area, and so the sampling points were 30 meters apart, while the active mining site is relatively bigger, and we sampled at 300 meters apart. The Balatok mines is even a bigger mining area, and we sampled again along the tailing ponds, this time at 500 meters apart. Also got the same parameters of the area, not just like in the case of Felix, the physicochemical characteristics also of the water. And we sampled 2 liters of water from each sampling point, stored this in an ice box, and brought them back immediately from Benguet to Manila to our laboratory here at the Institute of Biology in UP Diliman. And each water sample, was actually filtered using filter membrane with a diameter of 47 millimeters and a pore size of 0.2 micrometers. So this is supposed to be able to trap microorganisms. And using a vacuum pump, okay, so the microorganisms are supposed to have been trapped in the filter membrane, and the water samples were actually brought to the Research and Analytical Services Laboratory of NSRI for analysis of certain metals using atomic absorption spectrophotometry. For some specifics on what we did with the samples, so the samples were extracted, genomic DNA in particular, using a kit, a DNA Easy Power Water kit. And after extraction from each sample, we checked for the quality of the DNA that we obtained using agarose gel electrophoresis. And then the genomic DNA extracts were sent to Apical Scientific, formerly called the First Base, which is located in Malaysia, where paired and shotgun sequencing was done using the Illumina HiSeq 2500 machine. So once the sequences were given to us, the rest of the work involved a number of bioinformatics analysis. So we used different tools, and this is the bioinformatics pipeline that we followed. So Trimomatic was used to actually trim the raw sequences and remove the 5' and 3' adapters. And then FastQC was used to assess the quality of the trimmed sequences. We used a Fred value of 33. A Fred is actually a measure of the quality. It's also called the Q value, the quality of the bases in the sequences. And the higher the Fred value, the higher will be, or the better will be the quality. So we had a high Q value that was used. And then we used Metaspades. Actually, this stands for St. Petersburg assembly algorithm, which then assembled the raw reads. No? So we had, if you remember, the shotgun sequencing approach, which would have paired reads and whereby there can be overlaps that can be joined together to assemble the sequences. And MetaQuast was used to determine the quality, of the assembled reads. So once the reads were assembled now into contigs, then we used a classifier called Clark, which can classify DNA and RNA sequences. So in our case, we had DNA sequences, and then the sequences were matched according to their taxonomic assignments. These are just the results of the physicochemical characterization of the water samples. And from here on, I will be discussing the implications of the results as I go along. So if you will notice, in terms of the pH, generally, except for one side, the waters were actually alkaline in nature. In terms of the temperature, so there was lower temperature that was recorded at the Philex mining areas, well, because this was located in a higher or at a higher altitude compared with that of the Balatok mines, which had a higher temperature and was found at a lower altitude in Tuba and Itogon, respectively. In terms of the total dissolved solids, so we found more solids in the active site in the TSF3. TSF, by the way, stands for Tailing Storage Facility, compared with TSF1, which, as I have said, is already inactive and has been revegetated. There was also quite a high amount of TDS in the Balatok mines. And you can check correspondingly that the amounts that would be for the salinity and the conductivity would correspond with the amounts in the total dissolved solids. Having more, for instance, would then give also higher salinity, and because the ions present in the solids will be a measure of the conductivity, then there was also higher conductivity, as you can see in TSF3 compared with TSF1, and then again, Balatok being in the middle. For the metal analysis, when the results were given, copper is the main metal that is being mined in all the sampling sites, in all the mining sites that we went to. But we also wanted to find the presence of other metals, and so these were also analyzed. And as you can see in the results, actually there was none which was recorded in the TSF1 sampling site. This is, remember, the inactive site. Well, because maybe there was no more mining activity that is being conducted. And in the case of TSF3, there was also none which was detected. And this could be attributed to the efficient extraction of copper by the company, so that there would be no discharge that we were able to detect, that the AAS was able to detect in this tailing storage facility. Well, surprisingly, there was copper and other metals that were also detected in Balatok. While it is supposed to be abandoned, well, probably it was not cleaned when operations stopped, or this may be due to the stories of the locals whom we interviewed, that there were still small-scale mining activities that are being conducted by the local people. And probably because these are small-scale mining activities, the process is inefficient, and there could be a large discharge of metals along the tailing ponds. And so we correlated then these parameters to the Cyanobacteria and even the other microorganisms that we were able to detect. And the initial bioinformatics analysis, so this was what was given to us by Apical Scientific. The samples were coded, the lengths, the sizes of the reads, the yield, and so on. And first, we tried to visualize the diversity of the microorganisms using a Krona chart. Now, a Krona chart is an interactive visualization tool which shows you the different microorganisms that were detected. The ones in the inner circle would represent the higher order taxa, and those that are found on the outermost circle would be those at the genus and even species level. So we found a very large number actually of microorganisms. And because they were so many, we wanted to just concentrate on the most predominant types. And so in this figure, we tried to assemble the more dominant microorganisms, the taxonomic composition of these microbial communities using a classifier called Clark. And when the percentage abundance of the contings were matched at the phylum level, as you can see, the colored bars will represent certain groups. So there were many that represented the Proteobacteria, and then the Actinobacteria, the Firmicutes, the Bacteroidetes, and then the Cyanobacteria, and then other microorganisms. And well, just to check if indeed we were getting the correct information, because these are all theoretical algorithms that we were using, then we tried to scan the literature, and we found that these kinds of bacteria have also been reported in environments such as those that will be metal contaminated, both at the phylum level and at the genus level. For instance, Flavobacterium is said to survive in an alkaline, slightly alkaline environment and in the temperature ranges which we also found in our study. And then same with Enterobacterium and Pseudomonas, which have also been found in fresh tailings and mine sediments. But since my main interest would be the cyanobacteria, so what I did was to look at the contigs that would specifically correspond to the cyanobacteria. And so using default-based classifier of Clark, then these are the percentage proportion of the classified contigs in the main genera of cyanobacteria that were detected. And if you can see, actually in all the sampling points, all of these representative top 5 cyanobacteria were detected in varying proportions. But as you can see, again, following the color-coded bars, so the filamentous cyanobacteria, which would be Leptolimbia, these blue ones were found in all sites, just like also Nosto, which is represented here by the violet bars. And then the unicellular cyanobacteria were predominated by Synechococcus in the red bars, Cyanobium in the green bars, and Gliobacter in the light blue bars. No? I'm trying to prevent myself from using the pointer because it's moving to the next slide. So from here, we used a custom-based Clark to specifically zero in on the most dominant genera or species of cyanobacteria. And if you note, all the sampling sites pointed to Leptolimbia as the dominant species, or actually the dominant genus. Next, so what we did was to use a prokaryotic annotation tool to determine the genes that would be present. And this was through PROCA. So it's a prokaryotic annotation tool. And as you can see in the results, so there were a number of protein coding sequences that were detected, as well as genes for tmRNA. This is a bifunctional RNA transfer messenger RNA, and then genes for tRNA and rRNA as well. So we were interested in these protein coding sequences because we were thinking that this would be where then certain genes would be involved in metal tolerance. And so next, what we did was to now annotate further, and specifically now focusing on the metal response genes. This time by using another tool, which is called the EGNOG mapper. EGNOG, which stands for Evolutionary Genealogy of Genes, Non-Supervised Orthologous Groups. And as you can see again in the figure, again the color-coded bars this time will correspond to certain metal response genes. The top 5 metal response genes would be those represented by the pink color, which would be for copper, and then the green ones for zinc, and then the violet ones for silver, the light blue ones for iron, and the orange ones for cadmium. So at this point then, we were seeing that there was correlation in the ability of the cyanobacteria that were detected in the water samples with the presence of certain metal response genes, particularly copper, because as I've said, the mining sites were essentially extracting copper. Now, we actually used another classifier, which is called Kraken. We have another set of data for this, but in the interest of time, we just wanted to compare whether we were really seeing the same genes during our annotation, and we were not missing anything. As you will note, well, some sites showed the same metal response genes. In certain instances, 2 sampling points show this, and in some instances, only 1. So we were trying to balance and see to determine that we were not missing anything. And as you can see in this table, so there were a number of metal response genes, particularly for copper. Essentially those that will have to do something with the cation efflux system, which from literature is found to be the main mechanism for metal tolerance. The cyanobacteria are able to expel the metals from their system. And some other genes that will be responsible for the synthesis of certain proteins that will have something to do with homeostasis and transport and so on. Likewise, we also found other metal response genes for zinc, let's say arsenic, cadmium, mercury, calcium, and so on. So we were able to get a large number of these metal response genes. And just to show you the main difference of these tools, the 2 classifier tools are very similar, except that Clark can be run at a faster speed. That's why we resorted to this, because it's faster, and we were running so many sequences during that time that we were conducting the study. But noteworthy would be the fact then that Clark will be able to discriminate at the genus level, and that's why we had genera that were identified. And Kraken actually can discriminate up to the species level, but it is slower. So as I was saying, we were getting all this information from the algorithms that we were using, but we wanted to find out whether indeed such genes will really be present in the cyanobacteria, which would be our interest. So the next part of the study dealt with cultivating some of the cyanobacteria in the laboratory, and then trying to screen for the presence of these metal response genes and validate for their existence. So using gene annotation, we designed primers for those genes, and then after PCR, screened for the presence of the PCR product through agarose gel electrophoresis, and then this time submitted the amplicons to the Philippine Genome Center here in UP Diliman for Sanger sequencing for validation. Later on to match those sequences with, again, certain specific proteins, if indeed they are involved in metal response. So these are the genes that we tried to screen. So we designed, as I have said, primers for these genes, and then some other characteristics, amplicon size, and so on, that we consider the annealing temperature for PCR. So we had a number of genes that were run, including this another set of sequences. And we tried then to screen those genes in the cyanobacteria that seem to be promising. As you will note, Leptolimbia seemed to be the dominant genus, and we particularly cultured that strain that was isolated from TSF3. And then we also tested for Nodocylinea, which was actually isolated in TSF1. I was particularly interested in Nodocylinea because this has not been reported to be present in the local mining sites. And then we also screened for Cinococcus, which was isolated from Balato, because Synechococcus, being unicellular, is a very good test organism for synthetic biology nowadays. So we wanted to go into that in the future. And then we tested also for another filamentous cyanobacterium, which is for medium, also isolated from DSF3, which is the active mining site. So note that I was particularly interested in the lesser studied cyanobacteria. While we had detected and also cultured nostoc, say for example, or oscillatoria, I wanted to focus on the less studied cyanobacteria. So we did some morphological characterization and microscopy. And then so extracted DNA from the bacteria, cyanobacteria themselves, the individual cyanobacteria, and then, as I've mentioned, subjected the DNA extracts to PCR, and then we were able to detect, so these are just sample gels to show us the presence of, let's say, the COPR gene, the CSOR gene, which would be both for copper tolerance as well as the general SMTB genes. These are metallothionein genes, general for metal resistance. The minus signs represented the negative control, so there was no sample loaded in the lane there. And to summarize, because we had run many gels, we found that all the 4 representative strains were positive for the metal response genes that we screened and tested. And when the results of the Sanger sequencing came from the Philippine Genome Center, so we tried to match the sequences with the closest protein identification, and we found that these proteins would also be involved in metal homeostasis and metal metabolism. So to summarize and to give our future plans, so we found that metagenomic sequencing revealed the presence of cyanobacteria that can survive in mining sites. Bioinformatics analysis showed the presence of metal response genes, and the promising isolates, as I mentioned, were cultured and maintained in the laboratory at the Institute of Biology for further work on potential and actual bioremediation applications. And since we still have the sequences with us, for the future, we wanted to annotate for other genes, say for those that will be responsible for the production of bioactive metabolites for metal applications. We've read from literature that cyanobacteria are also good sources of anti-cancer compounds and even antiviral and antimicrobial compounds. So we would like to do this in the future, as well as probably try to mine for genes that could be used by the industry, like those that are responsible for pigments that can be used for the food industry and compounds that are usually used in the cosmetics industry, because chinorin is also a product of many cyanobacteria. And so just before I end, I'd like to mention that part of the requirements of this project will be the mentoring of graduate students. So we had 2 graduate students from the Institute of Biology who were able to finish their studies from this project. Mr. Angelo Joshua A. Victoria graduated in June 2018 for his master's in MS Biology degree. During the time that we were starting with this project, he was already at the thesis stage. And so what he did was to concentrate on one cyanobacterium that was already available in the lab, not those that we isolated in Benguet, but the Hapalosiphon that I initially isolated from a tailing pond in Marinduque from the Marcopper plant. And because we were just starting with the bioinformatics work in the laboratory, Mr. Victoria, through the project, was able to undertake bioinformatics training in Italy to complete the whole genome sequence of hapalocyte and complete the annotation of all the genes that are present in that organism. Now, for this particular lecture, the bulk of the data actually came from the work of Dr. Libertine Rose Sanchez, who completed her PhD in biology degree last year from this project. And as part of our final requirements, so we were able to publish part of the dissertation, the ones in Balato, of Dr. Sanchez. She's preparing another manuscript for the Felix mines. And lastly, I'd like to give the following acknowledgements to the Institute of Biology, College of Science. Particularly, well, the work was conducted at the Plant Genetics and Cyanobacterial Biotechnology Laboratory in IB. I'd also like to acknowledge our neighbor laboratory, the DNA barcoding laboratory, because we also used some equipment in their lab. And then again, the Philippine Genome Center for the sequencing of the Sanger sequencing of the DNAs from our representative Cyanobacteria. We also used the server of the PGC since we needed more computer storage space, and because we were running our sequences 24/7. Again, I'd like to thank the Natural Sciences Research Institute and DOST PSHERD for funding the project. DOST-AST-HRDP, which was the source of the scholarship of Dr. Sanchez. And this scholarship also enabled her to go to Germany at the European Molecular Biology Laboratory, where she undertook additional bioinformatics training so that she can assemble also and complete the data that I had reported through the European Molecular Biology Organization. And again, once more, I'd like to thank the management of Felix Mining Corporation for giving us the permit, allowing us to enter their facilities and collect water samples. Likewise, the LGUs in the towns of Itogen and Tuba, because we got permits from them so that we can sample in the areas that I have mentioned earlier. So thank you to all of these institutions, and thank you for listening.
Thank you very much, Dr. Cao. May I okay, there. So let's check. So thank you very much, Dr. Cao. So we often describe microorganisms as ubiquitous, and this is based not just on their metabolic plasticity, but also on their ability to tolerate different forms of stress. And as discussed, the identification of genes from microorganisms that can tolerate heavy metals is a first step towards using these microorganisms for bioremediation and other purposes. Now, for those who just came in, we will have the open forum after our third speaker. But if you have questions or clarifications for Dr. Rivera or Dr. Cao, you may type them in the chat box or Google form for those watching the webinar via YouTube. Now, for data privacy, all questions submitted for the open forum will be considered anonymous unless you type your name before or after the question. Now, our third and last speaker is Dr. Ian Kendrich Fontanilla. He is a Professor 5 and a UP Scientist 1. He obtained his PhD in genetics from the University of Nottingham, and he is the current director of the Institute of Biology and the principal investigator of the DNA barcoding laboratory to talk about the utilization of environmental DNA for Schistosomiasis japonica monitoring and biosurveillance. Let us all welcome Dr. Ian Kendrich Fontanilla.
Hello, good afternoon. Thank you for inviting me. And thank you, Dr. Pedullar, for that kind introduction. Okay, So my talk for today will focus on a particular disease that is quite prevalent actually in the Philippines, and this one is schistosomiasis. Okay, so my topic is the utilization of eDNA for Schistosomiasis japonica biomonitoring and surveillance. So just to give you an overview of our talk, of course, I will give a brief introduction, followed by the methodology, results, and conclusions and recommendations, as well as the next step, which includes an offshoot of our study, wherein we conducted another study in relation to our study, actually applying what we have discovered. And then after that, a way forward after this project. Okay, so just a brief introduction. Please indulge me for those of you already familiar with this disease. Schistosomiasis is a parasitic disease or infection caused by the body's reaction to the eggs of schistosoma. So what we see, the clinical manifestation of schistosomiasis, which includes anemia, malnutrition, learning difficulties, especially in children that are infected repeatedly, also liver damage to the liver damage, even the intestines, lungs, and bladder. Most of these are caused by our body's reaction to the eggs of schistosoma, rarely because of the adult worm. So here you have an example of a patient on the left, which has an inflamed or enlarged liver. And this one, the picture below, shows you a pair of schistosoma, the much shorter but stouter male, and the much slender but longer female. Okay, so these are the adult stages of the parasite. Now, Schistosomiasis japonica in particular is a big public health threat in the Philippines. It is caused by a particular species of schistosoma, which is Schistosoma japonicum, and it is endemic in 12 regions of the Philippines. In fact, in 28 provinces, even in 14 cities, 189 municipalities, and 2,221 barangays or villages. So there is an estimated 12 million Filipinos that are living in endemic areas. When we say endemic areas from the public health perspective, these are the areas where the parasites are found. And then around 2.5 million Filipinos are directly exposed to infection. Okay. So in fact, in as late as 2002 and 2005, we have discovered new endemic foci for this parasite. One in Gonzaga in Cagayan province, and the other one in Calatrava in Negros Occidental, shown here in the map. So here in Gonzaga and here in Calatrava. Okay. Now, just to give a brief overview of the life cycle of schistosoma, for those of you who are not familiar, so the schistosoma requires a snail intermediate host. So it goes through several larval or intermediate stages before reaching the adult stage. Okay, so this one is the adult stage, and as you can see in this diagram, the life cycle of the parasite requires fresh water as a crucial medium. So you have the eggs that hatch from a definitive or final host, which could be water buffaloes, humans, cattle, dogs, and others. And then these eggs hatch into miracidia, which then penetrates as a snail intermediate host, where the larval stage progresses into a sporocyst, and then the sporocyst will develop into a cercaria. Here in this diagram, you can clearly see the cercaria, which has a forked tail. Okay, that is a diagnostic characteristic of Schistosoma japonicum. And then this one burrows through the skin of a mammalian definitive host. So, and then where they will mature into adult in the definitive or final host. Now, Schistosoma japonicum or Schistosomiasis japonica is a true zoonotic disease, which literally means a transfer of the parasite from animals to humans. Okay. Now, the main intermediate host of Schistosoma japonicum here in the Philippines is Oncomelania hupensis quadrasi. Now, this one belongs to family Pomateopsidae. It's an amphibious and dioecious snail, meaning it can thrive in both aquatic and terrestrial habitats, but the sexes are separate. So the males and females are separate, unlike many of the land snails we see, wherein they are hermaphrodites. They have both male and female organs, sex organs. So these snails are relatively small, so they are only 3 to 5 millimeters long. So they're less than half a centimeter. And then if you're not familiar with these snails, if you burrow through the soil, you can easily mistake them for small pebbles or small rocks. So the shells are ovately conic with sharp apex. And based on our previous study conducted by one of our faculty at the Institute, Mr. Rocky Cornelius, they surveyed 3,012 snails infested bodies of water, wherein 80% are located in Mindanao, 18% in the Visayas, and 2% in Luzon. Okay. Now, how do we, from the public health perspective, how do we monitor and survey endemic foci? Because these are important strategies in order for us to determine where these endemic areas are and therefore take necessary actions or steps to prevent the infection to prevent human infection or transmission of the parasites to humans. So there are two approaches for surveillance. You have the human prevalence surveys, wherein you target susceptible segments of the population, including the school-age children and the working age group. So these usually they are found in areas where there are freshwater bodies where these snails can thrive. So snail surveillance in particular can also be done through malacological survey. Simply put, this simply means snail survey. So you determine the presence of the snail and the parasite in these sites. So how do you do that? You go to these sites, you look for these snails, and then you bring them to the lab, and then you crush the snails, and then you view them under the microscope to see if the cercaria will swim out. So if you can see the cercariae plural, swimming out of these snails, then then you are certain that these snails have been infected. Okay, so these are just pictures of some of the malacological surveys our team conducted in the past. So as you can see here, they will have to do this, or they had to do this in waterlogged areas where the snails are quite abundant. And these are how the snails look like. Okay, so if you're not familiar with the snails, you could easily mistake them for black, small black pebbles, or you can easily mistake them for because these snails also resemble some juvenile snails. Okay. So there are, however, problems with malacological surveys. So first off, it is very labor intensive. You'll have to scour the area for several hours, and then you have to involve a certain a specific team of individuals who are taxonomic experts in identifying the snails. And also, if these are endemic areas, they are also quite risky, these malacological surveys, because these malacologists also run the risk of being infected themselves. Okay? So dito ngayon papasok yung konsepto ng eDNA and DNA barcoding. This method utilizes fragments of the animal mitochondrial gene, in particular the cytochrome oxidase subunit 1 gene or COX-1 gene, for molecular identification of organisms. So ang ginagawa natin, we just get water samples or soil samples, and then we try to extract the environmental DNA present in the sample. So kaya siya tinawag na environmental DNA, you want to know they are present in the environment, whether as intact DNA in the individuals themselves, or they are slowed off DNA, or DNA released into the environment. Now we do high throughput sequencing or targeted sequencing based on the DNA extracted from here using polymerase chain reaction. Okay, and then through bioinformatic processing, we can eventually identify the species present in the environmental samples. Okay, so pag sinabi natin environmental DNA or eDNA, these are any genomic DNA released to the environment from a source organism from various matrices such as sloughed off cells, metabolic products, and tissue remains. So we can apply eDNA in various scenarios. You can use them for biodiversity assessment to determine what species are present in a particular area. And from the perspective of parasitological research, you want to know if a particular parasite is present without having to identify or seek out the actual parasite. Okay, So in fact, eDNA has been utilized in parasitology. For instance, it has been used to detect Toxoplasma gondii from soil samples, and also Cryptosporidium parvum in storm waters. And and then you have Opisthorchis viverini detected in freshwater samples. Now, eDNA was also utilized to a limited extent in schistosomiasis. For example, Driscoll et al. Utilized the retrotransposon gene in Schistosoma japonicum, so that's SJR2, tested on Schistosoma japonicum cercariae. So they made a lab simulation. They tried to detect uh, eDNA from cercariae in the lab. And then Hong and Remey used SJR2 and marine retrotransposon genes tested on field water samples that are spiked with known number of S. japonicum cercariae. And then Worrell et al. In 2011 tried to come up with modified sampling techniques from the environmental isolates. So what we hope to do in this study is to come up with an optimized eDNA detection system that specifically targets Schistosoma japonicum and Oncomelania hutpensis quadrasi. So we have to start it off from the laboratory, and then later on, we wanted to apply this in the field if they would work. So specifically, we try to design OH quadrasi and utilize S. japonicum specific primers based on the cytochrome oxidase subunit 1 gene and apply them to endpoint PCR and quantitative PCR setting. So there are already primers available for Schistosoma japonicum, but not in the context of eDNA identification. eDNA, environmental DNA detection. So we wanted to optimize the protocols for Schistosoma japonicum. And at the same time, we also want to develop primers for the snail intermediate host, Oncomelania hutpensis quadrasi. Okay, so to do that, we also wanted to perform specificity tests for the primers used against non-target snail and trematode species in an endpoint PCR setting. So we wanted to find out if the markers that we will use will exclude identification or will exclude other species other than our target species. Okay, so this is an overview of our methodology. So we had to design the primers as well as the probes that we will use for qPCR. And then water sampling. So yung una, laboratory water samples, we just wanted to show if our method would work. So we had to start off in a lab setting. And then eventually we tried to test it in the field using field water samples. And from both sets of samples, we tried to extract the eDNA. And then we conducted a specificity test, especially for the lab water samples using endpoint PCR. And then we conducted quantitative PCR assay of both lab and field water samples, on lab and field water samples. Okay, so for the primer design, so we used the COX-1 or cytochrome oxidase subunit 1 sequences from GenBank, all the available sequences, and we tried to align them and design a particular primer or probe that will target only Schistosoma japonicum. So, and then after that, we also have to conduct a blast and search of our primers, on our primers, so that we will test whether it will only identify Schistosoma japonicum and Oncomelania hutpensis quadrasi, or will they target other primers or other species as well. And then for the snail collection, this is for our lab setting. Of course, we needed to get some actual live snails from the field and bring them to the lab. So to test whether we can detect the snail DNA. So we obtained snails from a particular site, Gindagan Tanawan in Leyte, which is known for its low infection rate. So there are some previous studies that show that there are infections of the snail, but the frequency is was very low. So we tried to collect samples from there. And then we came up with an experimental ecotope setup. Basically, we tried to rear the snails in varying numbers in in containers that contain a fixed volume of water. So we tried to do that with 75, 50, and 25 snails. And then we wanted to extend it further, but that's another study, if we could detect DNA with just one snail. Okay. And then we have to, after that, we will have to mimic water sampling by filtering the water sample into a filtration setup. We install here a filter paper, Wattman filter paper, and through that filter paper, we will extract the eDNA. Okay. So for the field water samples, of water sampling, we targeted 3 provinces which are known endemic foci for this parasite. So you have Northern Samar, Leyte, and Compostela Valley, or Davao de Oro. And these are the municipalities and barangays that we targeted. And these are the codes we use for the samples, for the sampling sites. Okay. So for the field water samples, so we basically what we did was we had to do a two-pronged approach. We had to do the traditional malacological survey as well as the eDNA to determine if they will correlate with one another. So we did a surface water collection for our eDNA extraction, and then likewise we did snail sampling to determine if snails are in fact present there. And then we conducted laboratory water filtration, similar to what we did in the lab. And then for the Oh, and by the way, I forgot to mention, for some of these snails, we also had to crush them to determine if indeed they were infected with the schistosoma. And then we followed the typical method of eDNA digestion, binding and washing, and dilution. I won't go into the protocols, but these are already published in our paper, so you could get them. And then also in these, which we also based from this paper by Uchii and Minamoto in 2016. And then for genomic DNA extraction, we use a Quiogen DNA EC blood and tissue kit. Okay, both for the target organisms, Oncomelania hubensis quadrasi, the snail, and Schistosoma japonicum, and for the non-target organisms, which we had at the time. So we had several species of Limnea, Pomasea canaliculata, and Melanoides for the snails, and then we also use Fasciola, which also belongs to the same taxonomic class as the Oncomelania, as Schistosoma japonicum. Okay, and then we conducted endpoint PCR just to check if our primers work, meaning they can amplify a PCR product from our genomic DNA from our parasites. And then we had to do agarose gel electrophoresis to visualize gel extraction purification. And to confirm that what we got was correct, we had some of the PCR products we obtained, we checked for if indeed they were Schistosoma japonicum and Oncomelania hubensis quadrasi. So we had to do single pass sequencing. Okay, so this is what we use for our PCR. We use Tacman qPCR master mix and Tacman qPCR running conditions. Okay, so for our results, so we designed Oncomelania hubensis quadrasi primers for our eDNA identification. So true enough, we were able to target the primers that will zero in on Oncomelania hubensis quadrasi with 100% identity. For the Schistosoma japonicum, which are in fact previous primers, so we didn't have to design primers. We also double checked for their suitability as primer specific or species specific. So these are some of our results for specificity. So true enough, our primers that we designed for Oncomelania hubensis quadrasi using typical endpoint PCR did target Oncomelania hubensis quadrasi and did not amplify anything from the other snails. Okay, and also our Schistosoma japonicum primers, it did amplify Schistosoma japonicum, but nothing from Fasciola. Okay. So these are our profiles generated for the qPCR. Now, for most of you who are, I think many of you are familiar with the COVID testing, the swab test, wherein you do RT-PCR. Of course, we don't do reverse transcriptase PCR because we're not working on the RNA here. But the visualization of a positive result is similar. Ginagamit natin yung qPCR. Okay, so instead of running them in an agarose gel, we visualize them instead as profiles and peaks to determine if we are amplifying something. So we are using basically the same method or method or technology as what we do in the COVID testing. Okay, particularly the RT-PCR minus the reverse transcriptase stage. Okay, so ito yung aming results sa lab. So true enough, whether we have 25, 50, or 75 snails per 500 ml water, we were able to amplify or get PCR products from or amplify the eDNA of Oncomelania hupensis quadrasi. And nothing should be amplified from the negative control, which we also obtained. Okay. Now, interestingly, when we also when we also applied the Schistosoma japonicum markers on the same water samples, eDNA samples, we also amplified Schistosoma japonicum in some of these lab samples. Okay. So ito in 1 out of 3, 1 out of 3, ito 3 out of 3, yan. So uh, so itong mga samples na nakuha namin sa Tanawan were in fact infected with Schistosoma japonicum, and we were able to detect the eDNA from the lab. Okay. Now, for the field work, okay, ito naman ang napansin namin. So we first divided our data set or our samples into those wherein we did not observe any snails during the actual field work and those where we observed the snails. Okay, so ano yung lumabas sa qPCR? Okay, so interestingly, okay, so we did not observe any snails, but nevertheless, from the water samples, we detected the eDNA of Opensis quadrasi. So this is very telling in that although based on ocular inspection, you may not be able to see the snail, but in fact, some of these snails may be hidden or their eDNA are actually present. Okay, so these eDNA could have obviously came from these snails, which could have been there or which could have come from elsewhere. But basically, the eDNA of these snails were there. Okay. Now, interestingly, then, for those sites wherein we observed some snails, sometimes we also got a negative result for our eDNA testing. So this also tells us that, this also indicates the patchy distribution of environmental DNA. Just because the snails are present doesn't necessarily mean that the eDNA will also be homogeneously distributed in the site. Okay, so there is a caveat for environmental DNA testing. Now, for the Schistosoma japonicum, okay, interestingly, in the sites where we did not observe the snails, we even detected the eDNA of Schistosoma japonicum. Now, please remember that at least based on what we know to date, the Schistosoma japonicum cannot complete its life cycle without the snail intermediate host. So even if the snails were not observed, but the mere fact that we were able to detect Schistosoma japonicum seems to point to the very likely scenario that the snails were also present in the sites. Okay, and then here for those sites wherein the snails were observed, and then we tested them for infection date. So if we observed snails in a site, we also try to determine if we could get the parasite from these snails to test for infection date. So for example, 2.08%, so that's roughly 2 out of 100 snails are infected. Now for those snails that were not, for the population of snails from these sites, wherein we did not detect any infection, surprisingly, well, probably not surprising at all, we could also detect the presence of Schistosoma japonicum. So just because we did not detect the actual presence of the Schistosoma japonicum from our snails during the when we crushed the snails, the parasite could still be there. Okay, we could have just missed these parasites. Okay, so dito lang, just to give you some perspective, for ocular inspection negative, that's a fancy of saying for those areas where we did not find the snail, okay, so we were expecting them to be negative when in fact using qPCR to detect the eDNA, 3 out of the 12 sites were positive. For those that were Oncomelania hupensis positive, okay, just because we were expecting 7 sites to be positive, so they should also be positive. They should have been positive for eDNA detection, but only 6 out of 7 were tested positive. Now, for those Oncomelania hupensis ocular inspection positive for the presence of Schistosoma japonicum, out of the 5 sites wherein we detected Schistosoma japonicum, only 3 were positive for Schistosoma japonicum. Okay, so there seems to be a caveat here that just because you do not see the physical presence of either the snail or the parasite doesn't mean that there will also be a direct correlation with the eDNA, meaning that there should be any eDNA at all. So this points to the limitation of the physical search of the oncomelania and the schistosoma. So it's quite easy for us to miss these snails and parasites when in fact they were there in the environment as detected by their eDNA. And the opposite could also be true. Okay, so even if we see them physically, sometimes the eDNA test will still give a negative result. And this is precisely because there could possibly be many factors that will determine the resiliency of our eDNA in the environment. And plus the fact that the eDNA is not homogeneously distributed in an area where the source of the eDNA is also available or present. Okay, but based on our studies, we can successfully conclude that the eDNA utility and specificity using COX-1 or cyclooxygenase subunit 1 for S. japonicum and OH quadrasi has been successful. So the method successfully tested on field water samples, some of which tested positive for OH quadrasi eDNA and Schistosoma japonicum eDNA. And the method can be used to complement malacological surveys, and which is considered less risky. So ang kagandahan ng eDNA is that, of course, it makes it safer for our malacologists to just go to the field, collect water samples, and that's it, without having to immerse themselves into the field by wearing boots and then going to the water or going to the riverside or the river banks and then try to collect these snails. Okay, so our recommendations, we hope that we could rear OH quadrasis snails in the lab and can be utilized for in vitro experiments for eDNA under controlled conditions. So what determines the efficacy of the eDNA method? What conditions will make the eDNA be amplified? Okay, and for how long would eDNA last in the field? What other factors, environmental factors, should be considered? So these are wonderful experiments that we could work on in the lab under controlled settings. And then another question that is always been thrown at eDNA barcoding is that, can you determine the size or the population or the frequency of the individuals based on the eDNA? So, well, we can come up with a standard or calibration curve with amplification profiles to correlate the cycle thresholds and DNA concentration, and therefore provide quantitative data on the detection of target DNA from environmental water samples. But that remains to be seen. So what we just did was to detect the presence. We have not yet come up with a method to quantify the number of individual snails or parasites. Okay, so we have come up with a paper based on this one, and this one was largely part of the master's thesis of one of our faculty and a graduate student, Mr. Rafi Formilios. Okay, now because we were able to develop this method during the conduct of the project, and we still had time and we had enough money, we came up with an option of this project. Can we test this on soil samples instead of water samples? Because this could also be a very useful useful approach to determine the habitat range of the snail. How far can they go from the water where they will still, in order for them to survive and thrive? And then the presence of the eDNA and the snails or the parasites could also be correlated with environmental conditions. Because in fact, Oncomelania cupensis quadrasi is an amphibious snail. It can thrive in dry environments and aquatic environments. So we did that using with the help of our colleagues from UP Baguio. So they surveyed 3 villages in Gonzaga, Cagayan, which I mentioned a while ago was one of the new endemic sites, okay, or endemic foci. And we collected soil samples, okay, around 0.25 grams of soil sample. We extracted the eDNA, and then we used both conventional PCR or endpoint PCR as well as qPCR, okay. And at the same time, we tried to identify the edaphic factors of or characterize the edaphic conditions of the soil, per 500 per 0.5 gram of soil sample. And then based on that, we could detect the presence of these snails and then try to incorporate them in a GIS setup or geographic information setup or system. Okay, so based on that, we came up with another paper together with our colleagues from UP Baguio. So this study was a pioneering study that used eDNA to detect OH quadrasi in soils. Okay, despite the absence of what do you call this, an aquatic environment. So distances of recovered eDNA indicate that OH quadrasi can travel from 0.5 to 6.5 meters away from water bodies. And the recovery of eDNA increases as the pH, phosphorus, zinc, copper, and potassium content increase. Okay, so ito yung mga edaphic factors na kinonsider namin. And then distribution mapping confirmed that the snail can extend their habitat range at high amounts of elemental nutrients. In fact, these snails can hibernate, okay, so to speak, for extended periods of time until such time, especially during dry periods, until such time that the water comes back during the rainy seasons, and then they can, go back to an aquatic environment and then reproduce. So this study obviously has implication to the prevalence of schistosomiasis in a particular area. Okay. Now, as an offshoot of our project, we are now involved in another project called the foodborne helminthiasis problem in the Philippines, present status, response, and proposed intervention, in which we employ eDNA in the detection of certain parasites, including schistosoma, in animal samples as well as environmental samples. Okay, so way forward here, the way we see it is that rather than focusing on the classic diagnosis, we can have a One Health approach wherein we involve both the clinical approach and the ecological approach. For the traditional clinical approach, you detect the presence of the parasite based on diagnosis in human and animal patients. And then we can also use the ecological approach by zeroing in on the habitat or the environment of these parasites through eDNA and then incorporating the information or data in a GIS or geographic information system. And through these data, both eDNA and then classic and new diagnosis, we can have a multi-pronged approach towards One Health. Okay, we can target the parasite in humans, and at the same time, we can also detect their presence in the animal reservoir host, and ultimately incorporate the biology and the ecology of these parasites within the context of the environment, and therefore allowing us a broader perspective of how these parasites propagate and disperse by putting a block in their life cycle at some point. Then the parasite dispersal can be minimized or even prevented. But this can only be done through a One Health approach where various stakeholders and various experts are involved. Okay, so of course, I would like to emphasize that this project was able to facilitate the thesis of several students, foremost of which Mr. Rapi J. Parnillos, who completed his MS degree in biology in 2018. And then we also had three undergraduate students from the UP Baguio, Fritz Calata, Camille Caragian, and Julian Ella Mendoza. They were the ones who did the edaphic study, the soil samples in Cagayan. So I would like to thank several individuals, foremost of which Mr. Cornelius, and my collaborator, Dr. Lydia Leonardo, who is also a lecturer of the Institute of Biology and also affiliated with the UERM Graduate School. And then we also have Mr. Ian Quintabios of UP Manila, our clinician. Of course, Dr. Ernelea Cao and Dr. Kilang and Dr. Rivera, who were also involved in the thesis of Mr. Cornelius. Dr. Marcelo Sato from the Tokyo Medical University, wherein Rapi was able to conduct part of his thesis in his lab. And then we also have our long-term partners, Yuichi Chigusa of Tokyo Medical University and Megumi Sato of Niigata University, as well as Toshifumi Minamoto of Kobe University and Mihoko Kikuchi of Nagasaki University. And then, of course, we would like to mention also the help of Ma'am Imelda Gaspi of the Schistosomiasis Research and Training Center of the Department of Health office in Region 8. And then, of course, our colleagues from the University of the Philippines, Baguio, Fritz, Camille, and Jillian, as well as Professor Zenaida Bawanan. So thank you very much. I would like to thank, of course, the Natural Science Research Institute for allowing us to conduct this project. Also, the DOSTP shared through project grant number 1702. And of course, the University of the Philippines and the Institute of Biology and our partner institution. So marami pong salamat.
So thank you very much, Dr. Ian Fontanilla. So as mentioned, schistosomiasis is a neglected tropical disease that infects millions, and monitoring methods based on or environmental DNA in water samples can greatly benefit government efforts in controlling the disease. We will now proceed with the open forum. If you have questions or clarifications for Dr. Rivera, Dr. Cao, and Dr. Fontanilla, you may type them in the chat box or Google form for those watching the webinar via YouTube. Now, for data privacy, all questions submitted for the open forum will be considered anonymous unless you type your name before or after the question. So we'll now proceed with our first question. Okay, so our first question is for Dr. Wendell Rivera. This was sent via Google form coming from Mr. Von Philip Perez. Von Philip Perez would like to know, how can the Aptamer research on Aptamer be further improved, and what additional techniques would this require?
Okay, that's a good question. In fact, Aptamer is being applied into many fields nowadays. It is applied in environmental monitoring, what else? Of course, diagnostics, pathogen recognition. It can also be applied for cancer recognition. Remember that the Aptamer in itself can recognize small molecules, proteins, and cells. So with these characteristics, they can be applied in a number of aspects. The second part of the question asks what again? Sorry.
The second question is the second part of the question asks, what techniques would these further improvements require?
Well, it basically depends on the purpose. Say, for example, in our case, we would like it to be used for diagnostics. The platform that we were thinking is on the use of lateral flow assay. So that would involve, of course, the use of nanoparticles, of course, checking which membrane would be best to use for that application. So it totally depends on the purpose of the use of the Aptamer.
Okay, thank you very much, Dr. Rivera. Our second question is for Dr. Ernlea Cao. This was also sent via Google form coming from RR of Malabon National High School. RR would like to know if there are detrimental effects of cyanobacteria to the environment. So I think he means, do cyanobacteria have detrimental effects in the environment that might need evaluation? And if there are detrimental effects, what solutions can possibly be used to address this? Ma'am Ernie, naka-mute po kayo.
Sorry, I was muted. So hello again. There are actually many cyanobacteria which have been reported to produce toxic substances. These are what we call toxins, or yung mga of particular mention would be yung microcystis, which produces toxins. So there are many cyanobacteria also. How do we, let's say, remove them? It's actually quite difficult to do that because they are naturally occurring. And precisely that's why I'm not working on those microorganisms, not those cyanobacteria. But I don't know. I'm not working on those kinds, so I don't know if there's a way by which we can, let's say, remediate them or remove them from the environment. There are those that actually have been used, to produce, no, parang mga those that can serve as some kind of parang natural pesticides or insecticides. But I think the problem there is the fact that it's not very specific and it might damage other organisms in the same habitat. So, but yun.
Okay, thank you very much, Dr. Cao. So our third question was also sent via Google form. This is addressed to Dr. Rivera. Our participant would like to know what is the estimated commercial cost of the proposed Aptamer diagnostic kit for Trichomonas once the kit is perfected?
We don't have the answer yet because we haven't really finished the development of the lateral flow assay. But it should be much cheaper than the commercially available point of care assay that I mentioned earlier of another company. So probably you have seen from my presentation that it may seem very expensive, primarily because of the equipment and the complexity of the assays involved. But actually, once you have developed the aptamer itself and you are able to generate the right sequences, of course, of the aptamers that are specific to your target molecule, they will be much cheaper than your antibodies that you basically use for diagnostics.
Okay, thank you very much, Dr. Rivera. Our next question is for Dr. Cao. The question was also sent via Google form. Our participant would like to ask what specific throughput was used for shotgun sequencing and how much did this cost?
First question, it's actually 2 by 150 shotgun sequencing approach, which means that the DNA, the genomic DNA, was first shredded into small pieces, about 1 kb long, and then eventually assembled to form the contigs. How much? We spent 334,000 for the 9 samples, so it was about 30,000 something for each run of the sequencing. So relatively expensive, because if we do 16S, because we also do 16S for identification of the cyanobacteria, it's just 500 pesos forward and reverse primers already.
There's actually a follow-up question on that from the same sender. He asked, was bias correction included in the relative abundance calculation? And if yes, how was the correction performed?
Actually, in the bioinformatics tools, there were several steps by which we tried to make sure that the reads are of high quality. Now, in terms of bias, in terms of the percentage, so essentially the idea is that per, let's say, liter of the sample, how much of the specific contigs that will correspond to certain genera would be present. And then, so essentially the number will vary. And of course, there will be biases, and that's why there are many tools that can be used. And that's why we're not really in particularly reporting the percentage abundance of each one, because that is not my interest. I'm not a microbial ecologist. I'm more of a microbial geneticist. So what I just wanted to know is the presence of certain cyanobacteria, because if you have at least one cell, theoretically, then we can already propagate that. So I was not particularly concerned about the abundance in effect.
Okay, thank you very much, Dr. Cao. Our next question is for Dr. Ian Fontanilla. This was also sent via Google form. So the question is, how is it possible that eDNA for S. japonicum was detected in samples where there were no Oncomelania hupensis quadrasi?
Thank you for that question. I think the most logical answer to that is the snails were present, but we did not find them. So because the Schistosoma japonicum cannot complete its life cycle unless it utilizes a snail intermediate host. Now, another possible explanation is that maybe it's using another snail as an intermediate host. But so far, based on our studies here in the Philippines, we only know of Oncomelania hupensis quadrasi as the intermediate host. So they are very, very specific to Oncomelania hupensis quadrasi. So that's the limitation of the malacological survey. You may miss them because they're very, very small. Ganun na sila kalito. So it's quite easy to miss them, especially if you're not, if you're going to survey a large area and you're going to do either convenient sampling or purposive sampling. It's quite easy to miss certain areas. So the simplest answer is that the snail was there, but we did not see them.
Okay, thank you very much, Dr. Fontanilla. The next question is for you as well. One of our participants would like to know if the technology you just described can be used for human samples, like saliva, urine, and feces.
I see. Yes, we can, but the thing is, there are other methods that we can use to diagnose a patient, whether the patient has schistosomiasis. In fact, one of our partners developed an ultrasound method. You just use a portable ultrasound, pinapadaan na doon sa liver, and you can actually see if there are lesions that are caused by the parasite. So yes, you can, but it may not be practical because diagnosis of schistosomiasis is quite straightforward without having to use molecular markers. Yun.
Okay, thank you very much, Dr. Fontanilla. One question we got from the chat box, so very interesting researches. So he asked, for someone who finished BS Biology 12 years ago but did not go to MS or the PhD route, would there be any chance for someone like him to join any upcoming research to be an RA for career switchers? Would anybody of our speakers would like to answer that question?
I'll go first. I think yes, you can. But if you're going to work with us at the Natural Science Research Institute, most of the time the projects will require that the research assistant is also enrolled in a master's or graduate program. So yun lang naman. But graduate studies is fun. Why don't you try it?
Okay. How about our other speakers? Any response?
Well, you can apply for graduate studies at the Institute of Biology, and Well, because usually yun nga, we want our graduates, our RAs also to be graduate students. And we can assign you offshoot projects. For example, in my case, I have two MS Micro students now, whom I have assigned the leptolimbia and nodosolinea respectively. So they will be working on the entire genome characterization of those two genera, since metal tolerance nga lang yung inaral namin to start with. So we have other strains that we can ask you to work on also.
Okay,
So
thank
that's
you.
for my part.
Okay, how about Dr. Rivera? Any response to that question?
Well, of course, there are always opportunities in research, especially in UP Diliman. Dr. Scow and Fontanilla mentioned that one of the requirements would be for you to become a graduate student, probably in the Institute of Biology. So since you're a BS Bio graduate, as early as now, it's best for you to identify your research interest. My field of interest is on microbiology, in particular, protozoology and applied bacteriology.
Okay, thank you very much to our three speakers. So please take note, if you're interested in joining any labs at the Institute of Biology or in NSRI, you are required to pursue graduate studies as well, not just to do research. So thank you to our panelists. Now, our next question is from Dr. Mo Vega from MSEP. She asks, at which stage in the life cycle of schistosoma does it become pathogenic? If schistosoma eggs are present but no host is present, is it assumed that it can already cause the disease?
Thank you, Mo. Hi. The egg is released from the mammalian definitive or final host, which includes humans. So it is released into the environment, and then it proceeds to the snail. Now, the infective stage of schistosoma to humans is the cercarial stage that emerges from the snail. So it infects humans by burrowing through our skin. And then after that, it progresses to the adult stage. And then it will go eventually to the liver and in other parts so that where they will lay their eggs. So the egg stage is the pathogenic stage for humans because of our immune response to the presence of the eggs. So ang tanong mo kung parang mahirap na mag-progress yung schistosoma to the egg stage if it is not in a definitive or final host. So it must find a definitive or final host in order for the adult to lay the eggs. Yun lang.
Thank you very much. Our next question is also for Dr. Fontanilla. This question was sent via Google form. The participant would like to know what factors contribute to the distribution of schistosoma in our country.
Actually, isang palaisipan yan para sa amin kasi the schistosoma requires the hosts. So where the hosts go, so will the schistosoma. So if you have the mammalian host, humans, cattle, pigs, dogs, wherever they go, if they are infected by schistosoma, then the schistosoma will go where these mammals go. And then if they're released into the environment, they will also require a snail intermediate host. So if Oncomelania is present, then the presence of the nematode will also persist in that area. So the next question would be how did Oncomelania hupensis quadrasi get here? Based on our studies, we have Oncomelania hupensis, which is widely distributed in Asia. So, and we don't know how old this species is for how many millions of years, even the genome. So, ito ay isa pang magandang study that we are going to evaluate how Oncomelania get to the Philippines and how did Schistosoma get to the Philippines, because Schistosoma has a widespread distribution, especially particularly the genus. So it is found in Africa, all the way to East Asia and in Southeast Asia. So yun lang, the parasite will be dispersed depending on the dispersal pattern of its intermediate and definitive and final hosts.
Okay, thank you. There was a follow-up question. How
Okay.
long can environmental DNA stay in the environment, whether in the soil, water, or air?
How long? Well, I can only speak for schistosoma. Uh, you know what they did? You know what they did in Japan? They tried to eliminate schistosoma japonicum via the eradication of the snail. So how did they do? How did they do that? They eradicated the habitats where these snails could reside. So they modified the environment. So instead of using carabaos or livestock or farm animals, they use mechanized, for example, tractors, etc. So that will minimize the infection, the transmission. And then they cemented the drainage ditches. So they did this for several decades and are successful in doing it. So if we want to eradicate schistosoma, we have to modify the environment. And then if they go for several dry seasons for extended periods, that will spell the end of the snail. And if the snail disappears, then the parasite will also disappear. So for in our experience in the Philippines, they can go through the usual season of wet and dry. So they can go through a dry season. Now, but I doubt if they will if they will live longer than one dry season. So baka pag tumagal pa doon, mamamatay din yung mga snails. And with it, mamamatay din yung mga parasites because their life cycle will be interrupted.
Okay, thank you very much. Our next question is for Dr. Wendell Rivera. This was sent via Google Form. Can we use the Aptamer technology for vaccine development or in conjunction with vaccine development?
One function of the aptamer is to block recognition because it's able to recognize a particular molecule. So in that case, it can block. So if that is the basis for your vaccine production, that can be used. Aptamer can be used for that purpose.
Okay, thank you very much, Dr. Rivera. So that will be our last question for the open forum, given the time we have. Thank you very much to our participants for actively engaging with our speakers. And of course, thank you to all of our three speakers, to Dr. Rivera, Dr. Cao, and Dr. Fontanilla. Now, for questions that were not read during the open forum, the seminar committee will forward them to the speakers who have agreed to answer them by email. Now, to show our appreciation to our three speakers, may I request Dr. Rivera, Dr. Cao, Dr. Fontanilla, and Dr. Jonas Kilang to turn on their video cameras for the virtual awarding of the Certificate of Appreciation. So the citation reads, the Natural Sciences Research Institute, University of the Philippines, Diliman, Quezon City. Certificate of Appreciation is hereby granted to our three speakers, Dr. Wendell Rivera, Dr. Ernelea Cao, and Dr. Ian Kendrich Fontanilla for delivering their talks entitled Aptamer-based Assay for the Detection of Trichomonas vaginalis, Metagenomic Sequencing of Cyanobacteria from some mining sites in Benguet Province for genome mining of stress tolerance genes, and utilization of environmental DNA for Schistosomiasis japonica monitoring and biosurveillance during the NSRI webinar series with the theme Genomic and Molecular Tools for Biosurveillance of Bacteria and Parasites, held on October 30, 2020. Signed, Dr. Jonas Kilang, NSRI Acting Director. So once again, thank you very much to our speakers. Now, some announcements before we have the closing remarks. First, there will be a virtual photo op at the end of the program. So participants will be asked to turn on their video cameras while a member of the seminar committee takes a screenshot of our images. Now, the pictures will be posted in the website and FB page of NSRI together with the video recording of this webinar. Now, second, participants who would like to receive a certificate of attendance are required to accomplish an evaluation form by clicking the link shown on the screen in the chat box. Now, take note that the link will close after 1 hour. Now, this event would not be possible without the hard work of the NSRI Seminar Committee. So, to give the closing remarks, let us all welcome its chair, Ms. Erna Elias.
Thank you, Dr. Pinulyar. Good afternoon, everyone. On behalf of the NSRI Seminar Committee, I would like to thank our esteemed speakers, Dr. Wendell Rivera, Dr. Ernalea Cao, and Dr. Ian Fontanilla for sharing with us today their fascinating lectures and their researches that I hope everyone was inspired by. And then thank you as well to our MC and moderator, Dr. Hil Pinulyar. I would also like to thank the NSRI Acting Director, Dr. Jonas Kilang, for his contribution and support in organizing this webinar. And of course, thank you so much, everyone. Those who were able to attend and those who have participated in the open forum, thank you. We hope that you have learned a lot from the lectures this afternoon. And we hope to see you again in our future webinars. So please visit the NSRI website and Facebook page for announcements. Thank you. See you next time.
Okay,
Back to you, Samuel.
thank you very much, Ms. Elias and the NSRI Seminar Committee. We will now proceed with the virtual photo op. So please turn on your video cameras. So we just some reminders regarding data privacy. So those who prefer not to turn on their video cameras may do so. And those who prefer not to be included in the virtual photo op may leave the Zoom meeting, but please don't forget to accomplish the evaluation form. Thank you.
Okay, hello everyone. So with your permission, I will screen grab the Zoom gallery for documentation of this webinar. So kindly give me about a minute or 30 seconds to 1 minute. So 4 pages. Okay. Okay, thank you.
So once again, thank you very much for joining our webinar this afternoon. We hope that you learned a lot and now appreciate the value of genomic and molecular tools in the detection, monitoring, and biosurveillance of bacteria and parasites. If you have other questions or clarifications about the topics discussed today, you may contact our speakers. Their email addresses are indicated in the website of the Institute of Biology. So the program is over. Have a great afternoon. Stay safe, stay well, and stay healthy. Thank you.